Welcome to the Big Idea podcast, where we focus on food. Today we're going to be talking to Mohammad from Bright Biotech. Bright Biotech are a company based in the United Kingdom, focused on some really interesting food-based technologies that will help unlock the cell-based meat industry. So let's get into the conversation. Look forward to getting your feedback at the end. Thank you. Okay, welcome to the Big Idea podcast, Mohammad, where we're focused on food. How are you doing? What's new?
Hi, Andrew. I'm doing so well. A lot better than last week. I had to fight COVID for 2 weeks, but today I'm doing so well. So very grateful that I've managed to not suffer a lot. I know that a lot of people have suffered from COVID.
Well, I'm glad you're feeling better. I was nervous when we said we were going to talk last week and then you had to cancel at the last minute, but I'm glad you're feeling able to have this conversation today. So let's jump into it. Tell us what you guys do. Let's start there.
Great. So I'm the CEO of Bright Biotech. It's a company, we are a UK-based company. We registered the company in end of 2019. And that was just like a few months before COVID started. But the company is based on research that has been ongoing since almost like a decade. And I was part of it. I came from Lebanon in 2011 to the UK, and I joined the lab of Anil Day at the University of Manchester. And the technology is all about exploring the plant as a vehicle to make recombinant proteins. Now, to me, that was very exciting. The first time I came across the plant system as a vehicle to make proteins was when I was doing my undergraduate in Lebanon. And there they were talking about the more established systems that are used to make proteins for therapeutic uses. And we just came across, we scratched the surface of the plant system. So I did my homework and I did further research. And I felt that there's got potential to use this plant system in order to reduce the cost of making recombinant protein, whether it is for industrial uses or therapeutic uses. And that's why I ended up here in the UK. And in those 10 years, I've been trying to optimize the system and to get it to a stage where we can commercialize it. And this is where we are right now.
Oof. Yeah, did you take a breath there? I'm like, okay, there's a lot to unpack there. So yes. You talked about using plant systems as a vehicle or as a process for creating recombinant protein. Why don't we kind of unpack that for people who are sort of coming to this category or this space for the first time? When you talk about plant systems, you are not talking about production plants like factories and related, are you? You're talking about regular green plants, stalks, stalks in the field type plants, right?
Exactly. So we're talking about real plants. And in our case, we're using tobacco. I know like a lot of people, they run away when they hear the word tobacco because they associate it with bad habits. But then there's an alternative use to tobacco. We can use it as a cheap bioreactor to make proteins. And in our case, we're genetically engineering those plants to make animal proteins so they can be applied to a number of industries. So answering your question, it is the green plants that we are using.
Okay, so, and then you threw a few more terms in there, which I want to make sure people are clear about what they are. So you're using green plants, you're taking proteins, you're basically putting them inside of the plant, inside of the tobacco plant. As the tobacco plant grows, it basically multiplies or makes the protein that you put inside the plant grow, duplicate. So let's say, for example, and I'll be super simplistic about this, let's say you put 10 protein cells inside of the plant. As the plant doubles in size, you expect those 10 protein cells to become 20 protein cells if it's doubled in size. Totally not the case in so many different reasons why. But that's the basic idea, right? That as the plant grows,
Yeah,
the protein that you put inside of it grows too.
Yeah, so it's slightly different. So basically what we put in is not the protein. We put in the code that, so that encodes the protein, basically. So we go, for example, in our case, we're making now growth factors for the cultivated meat sector. We go to the animals and we go to the sequence of those growth factors. We introduce them into the plants, into the genetic system of the plants, and then we use the system, the genetic system of the plants, in order to amplify, in order to make those proteins, and then harvest them eventually. So so it's more or less.
So all of the scientists who are listening to me describing the simplistic view were laughing their rear ends off at how I was describing it, I'm sure.
Well, you got some of it.
That's good. That's good. I always love getting on these calls because I'm always the dumbest person in the room, which allows me to ask the stupid questions, and that helps us to understand. I will continue to take that role of dumbest person in the room, I'm sure.
Well, I realize there's never like a stupid question, because in my case, I used to be afraid of asking questions, and then I realized that asking questions is the only way to learn. I used to I don't know if you'll get time to talk more about myself, but I always used to identify myself as an introvert. And then I used to I take note of everything I hear, and then I never ask the question in order to clarify. So I used to do the research myself, and it used to take me so much time to find the answers. But then I realized in the process of building Bright Biotech that the fastest way is to ask questions, even if you think it's stupid. There's no stupid question.
Okay, well, I guarantee to ask the stupid questions. How's that? so Bright Biotech.
Yes.
Let's dig into it, and I'll continue to ask those silly questions. Tobacco.
Yes.
Is it because it's a particularly fast growing plant? Is that why you're choosing that?
So there are a number of reasons why I've chosen tobacco. So one of them, tobacco is a very high biomass plant, and then our system is a leaf-based expression system. So the more biomass you have, the more protein that you're producing. So this is one of the reasons. Another reason, it's because of the tobacco is a non-food crop. And then one of the reasons that slowed down the adoption of the plant technologies, especially the competing ones, is the issue of using food crops or the contamination of the food chain by the escape of the gene that you're introducing into your plant genetic system. So using tobacco is a non-food crop, and it eases the concerns about the contamination of food chains. So this is another reason. The third reason, tobacco produces a lot of seeds. So when we do the process of genetic engineering, and then we get our genetically engineered plants, this one plant gives us 10,000 seeds. So this relates to the scalability of the system. So it's because of these three reasons that we've chosen tobacco, and the levels of expression that we've seen in tobacco compared to other systems or crops that we've explored is a lot higher. So it's a number of reasons that made us choose tobacco over others.
Did you consider other crops?
Yeah, well, part of my PhD was looking at lettuce, for example. So so we did consider other crops, but the yields that we're getting from tobacco is a lot higher. And the system is more established. So the system in tobacco is a lot more established than the other systems.
What does that mean?
So it's been studied for a number of times. So we're not the only people in the world that have explored chloroplasts, because this is one thing that I didn't mention. So within plant biotechnology, there are three systems. So you can introduce the foreign gene or this foreign sequence that is coming either from plants or coming from any other living organism into the nuclear DNA of plants, or you can introduce it into the DNA of chloroplasts. Now, for the people that don't know, chloroplasts are the organelles that are present within the plant cells, and then they're famous for carrying out photosynthesis, which is the process of converting light energy into chemical energy. These organelles, they have their own DNA, and then they have them in very high copy numbers. So we introduce these foreign genes into the DNA of the chloroplasts, ending up in a very high copy number, allowing us to produce large amounts of the protein. So these are the different plant systems. And then you also have a third, which is a transient, where you introduce every time you want to produce a protein, you introduce the foreign gene through a bacteria that you introduce into your plants. Now, in our case, we're using chloroplasts. That gives us an advantage compared to the competing systems.
Now, in terms of growing this tobacco in large numbers, do you anticipate ultimately having a field of your genetically modified tobacco plants as a way of creating the volume you're looking for, or are these sort of kept in sort of internal container type growing systems, and you'll sort of increase the volume through multiple internal protected environments? I guess I'm wondering a little bit about the possibility, as you mentioned before, of sort of cross-contamination between your tobacco plants and other people's crops next door or whatever it might be.
This is definitely what we are hoping for, to be able to grow the plants in the fields, because everyone that talks about the plant biotechnology, they say that this technology will achieve its potential when the plants are grown on the fields. But then, as you've mentioned, there's a regulatory concern of the foreign gene escaping, and this is what puts us ahead of the competing plant technologies. Now, the advantage of using chloroplasts is that the gene will be contained within the chloroplast, and it will not be inherited through it will not go into the pollen grains, as opposed to introducing the foreign gene into the nuclear DNA. So once it's in the nuclear DNA, it goes into the pollen grains, and then it can spread through the fields. In our case, the chloroplasts are maternally inherited, meaning that pollen grains will not carry the foreign gene, and this will limit the spread. So we've got the potential to grow these plants in the fields. We are ahead of the competition in that terms, but there's no yet a regulatory framework that allows us to do that. So as a starting point, we're going to grow them in contained glass houses, but then the next step is to grow them in the fields, because this is eventually will allow us to even further reduce the cost of making the protein.
You said limit the chance of cross-contamination, limit or make it non-existent?
Non-existent. So yeah, so that's the right word. So it's a non-existent, because if it's not present within the pollen grains, it would be very difficult for it to escape into the environment.
Very difficult or impossible?
Again, so yeah, so it's the thing is, there are certain, like if I want to go into the technicalities of the technology, there are certain stresses that allow the pollen grains to carry a little bit of the chloroplast, but this is very, very, very rare. So that's why I'm telling you, up until this point, these need to be raised to the regulatory authorities to look into that. It's very rare, but then if you look at the biology, there are certain stresses, for example, very, very low temperatures, that where you end up with the pollen grain carrying the chloroplast. But usually you grow your plants at the optimal temperature, so around 25 degrees Celsius. So at optimal temperatures, it's not, so there you do not have carryovers of the chloroplast into the pollen grains.
So if you, for example, created a field
Yes.
where you're using your plants, is the intention to use the tobacco to extract the proteins from the plant,
Yes.
and then use the tobacco part of it for tobacco and the protein part of it for protein? Or has the process of creating those plants the way you want them to be made them monofunctional? In other words, they just are all about the proteins and they're not about the tobacco whatsoever. It's just a vehicle to do what you want to do.
This is actually a very interesting question, because it's one of the things that we've looked at. At this point, we're just using the leaf material in order to extract the protein, and then everything goes to waste. But then if you look at the protein content of tobacco leaves, which are considered to be nutritious, they have, besides the protein that you're making them make, they have endogenous proteins, and they account for around 20 grams per kilogram of leaf material. And then in the alternative protein industry, these could be used. So
is this the Rubisco aspects or something?
Many proteins. So yeah, so Rubisco is one of the proteins that are present. So you have like a whole range of proteins that are produced within the plant cells. So, and then as I told you, they account around 20 grams per kilogram, and that's quite a lot. So instead of wasting all of these 20 grams, we were thinking of looking at a side stream where we use this byproduct and also in the alternative protein industry. The thing is, one of the things that we need to look at is the nicotine content, because one of the drawbacks of using a tobacco plant is that they've got like a nicotine content. Now, in our case, we do have access to seeds that are low in alkaloid content, which is below the threshold. But then with the advancements in technologies, you can always shut down the biosynthetic pathway and then just make an alkaloid-free tobacco plant, and this would be ideal. And then if that's the case, then you can use this waste material and use it in several industries. So it's actually, a very interesting thing that we thought of.
I would recommend you talk to the Good Leaf company, which is a company in cohort 5, the latest cohort, because those guys are literally focusing on extracting proteins and converting that protein into a flavorless, fragrance-free protein source using leafy greens. So they're focused on the Rubisco side. They've been using sweet potato and other plants as the kind of core ingredient. But if you guys are going to end up with bunches of tobacco side streams,
yes,
this is what they've been focused on from a university perspective now for quite a few years as well. So maybe there's a collab here.
Yeah, that would be really interesting to speak to them. And it's good that you mentioned Rubisco. So Rubisco is the most abundant protein on Earth. So if you look at the protein fingerprint of the proteins present in cells, you see like on a protein gel a very thick band. And then our system is capable of producing a foreign protein to comparable levels to the Rubisco. So this is how good our system is. So
amazing. Amazing. So this company is based in Australia, so you'll have to figure out the time difference, but I'm sure we'll introduce you and you can have some of those conversations. So now let's get back to Bright Biotech. Yes. So we've kind of talked a little bit about the plants. I feel like we've got a grip on that. Now you're using that plant to ultimately create a growth factor.
Yes.
Do you have so for those that don't know, take us through what that growth factor is typically used for and how you see it being used in the food industry?
Great. So just like a background about growth factors. So these are naturally occurring proteins that are produced by cells, and then they promote a number of biological activities, including the multiplication of the cell, the growth of the cells, and then the differentiation into different cell types, if we're talking about stem cells. So these growth factors, they can be also used in a number of industries, including, as I said at the beginning, in regenerative medicine. So they have been used in tissue engineering and organ engineering. So they mediate or they drive the growth and the multiplication of cells. Now, cultivated meat industry is a derivative of the regenerative medicine. So you need to start off by a few cells, and then you need to amplify those. You need to grow them, and you need to allow their multiplication. So you need growth factors to be present in the culture media that they are grown in. And this is what makes it very difficult to scale this industry, because growth factors are not readily available at this point because they are difficult to make. And they are quite expensive. So this is where we can come in, because we are putting our technology on the table, a technology that can produce them at large quantities and also at a very low cost and a very scalable system. So we will be able to produce the amount of the growth factors that is required that will allow the industry to scale. So, and growth factors, as I've mentioned, so they are components of the culture media where the cells are grown.
So just to do my idiot summary again, the growth factor is used to make cells multiply. Growth factors are being used in regenerative medicine, i.e. to kind of, anyway, I'll let you define regenerative medicine. I'm about to completely butcher that. Regenerative medicine and also in cultured meat. So as a way of making the cultured meat cells grow. Typically that growth factor is difficult to acquire and is also very expensive, which is one of the key blockers or bottlenecks of scaling up the cultured meat industry.
Exactly.
And you guys have through your technology created a far less expensive and potentially more ubiquitous source of growth factors, unlocking cultured meat as an industry, as a category.
Exactly. Yeah. So yeah, everything you've said is spot on.
Whoa, that's a first. Perfect. So question, as effective, less effective, more effective than traditional growth factors?
Okay, so so let me highlight here that when we started exemplifying our technology with the growth factors, we started off by putting in human growth factors into the plant system. So the growth factors that we have expressed are human growth factors, but that doesn't mean that they cannot be used in the cultivated meat sector, because you're not taking anything from humans, you're just taking the sequence. The most important thing in this industry, you want to have potent growth factors. So this is what's important. The thing is, we've tested the bioactivity of our growth factors, comparing it to commercially available growth factors, and it showed higher potency. And this is very important because when you have higher potency, it means that you don't need as much growth factors in order to achieve the same yields, whether if we're talking about proliferation and the multiplication of the cells. So the way to move forward is to test the growth factor that we have on species-specific cell lines. And then the next step for us is to expand our product portfolio to include species-specific growth factors. And this is what we're doing now. So in the past month, as part of the Big Idea Ventures accelerator program, we've been talking to a number of cell-based meat industries, and we've heard from them what are the growth factors that they are interested in, what cell lines they are working with. And based on that, we put a list of all of the growth factors that we will start introducing into our plants, so we can have them available by the end of the year, so for them to start testing on their cell lines.
So again, I've got follow-up questions. You talked about using human growth factors. Let's say, for example, you take a piece of beef or some beef cells, and you grow those cells. Once the cells have grown, the growth factor is no longer part of it's not part of the beef. In other words, the beef that's created, or the cells created, won't include any component of the human growth factor that's used to multiply those cells, or will it?
It does depend on the growth factor that you are using. So there are certain growth factors that are uptaken by the cells,
okay,
and others that are not uptaken. So so it's so the right answer is that it depends on the growth factors. Some of them, they will be uptaken. So if we are starting off with a human growth factor, you might have traces within the plant cells, and this is part of the regulatory framework that needs to be in place. So one of the questions that need to be answered by all of the people in the cultivated meat sector is whether or what are the levels of the growth factors or the exogenous growth factors that have been uptaken by the cells.
Because I'm not sure many people are going to want to eat beef that is including human.
Yeah, so that's why in our case, we're moving away. So basically, we have exemplified our system with the human growth factors. But then, because initially there was a pivoting in the business model, we were a medtech company. And then just after we were introduced to the cultivated meat sector, we just shifted away from the food, from the human growth factors. And now we're going to be making species specific.
Now, maybe we shouldn't be squeamish about the fact that there's human growth factor involved, because to your point, it's the sequence or the code. It's not actual,
it's not a human, because you can look at it as a synthetic sequence. Because even, for example, let's assume we take a bovine, which is a beef growth factor, and then it has its limitation, whether it is in terms of stability or in terms of activity. So you can always make tweaks to the sequence in order to enhance its activity and enhance its stability. So it's a synthetic sequence. And basically, in our case, also in the process of introducing it into the plant, we take the protein sequence. You know, the protein is made up of building blocks, and these are amino acids. And then in our case, we don't change the sequence of the amino acids, but when it comes to the bases that encode the amino acids, we do changes in order to make it compatible with the plant system. So you can, like, it is the human sequence, but then it's a synthetic sequence that we've optimized in order to get higher levels. So think about it like evolution. Evolution, sometimes you get like mutations, and then if it's a favorable mutation, it remains. So in our case, we call it a human just because the sequence is a human, but it's not actually human.
The way I'm thinking about it isn't as a mutation, because that's one of those scary words that non-scientists kind of will probably freak out about, me included. I think of it as a recipe book. So you're using a particular recipe, which is telling you, like, it's a code, it's a recipe, it's a formula. It's not actually the bits and pieces from the human.
Yeah, exactly. Yeah, but I think people shouldn't associate mutation always with a bad thing. Sometimes you get favorable mutations, and This is what allows us to survive.
I'm just sort of thinking about the cultivated meat PR engine, which is getting the rest of the world, getting the rest of the world behind the category as a feasible, non-scary thing that you can feed your kids on a daily basis. And to do that, we need to be kind of cognizant of what sort of phraseology and so on will make alarm bells ring, even though they shouldn't. So from a calculation perspective, have you sort of figured out, given that the cost of the traditional growth factor has been a key barrier, what's your calculation of using your new approach? What's that going to do in terms of reducing costs? How accessible now is this is cultivated meat, given that of what you can achieve now?
Yeah, so I'll tell you about the cost that we have for example, the protein that we've made in our system. The retail price is $80 million for 1 gram of the protein. So it's massive. So in our case, our manufacturing cost is $100,000 US. So that's a big difference. And then because we want it to be a win-win situation for the company in order to keep innovating for the cultivated meat sector, and also a win situation for the cultivated meat sector to scale up, we are thinking of selling those growth factors a gram for $600,000. So we're talking about $80 million US versus $600,000 US. So in terms of the price of the cultivated meat, at this point it stands around a kilogram of cultivated meat around $10,000 US because of the high cost of the growth factors and the growth media. Now, introducing or replacing the current growth factors with a plant-based one, we're hoping that we can reduce the cost from 10,000 to all the way down to around 500, which is still very high. But then there are a lot of variables that all need to be optimized in order to get to where we want to be. But this is just like a big step forward towards reducing the cost of cultivated meat, in order to allow it to reach at one point cost parity with conventional meat.
So in terms of the product you're attempting to replace, are you attempting to replace FBS, or are you attempting to replace an alternative to FBS that's currently used in its place?
So at the starting point, we're just now replacing the growth factors in the non-animal media. So non-animal media is supposed to replace FBS. And then there are a lot of drawbacks of using FBS. So besides the unethical way of obtaining the FBS, there's also variability from batch to batch. So the best way is to replace it with a non-animal media. And then the non-animal media will have to include certain components. And then the growth factors are the major cost drivers in the marginal cost of this media. They make up actually 95%. So Yeah, now we're expanding our portfolio to include all of the growth factors that should be part of this media. And at one point, yeah, why not? We can think of actually making the media itself.
So when you talk about 1 kilo, did you say 1 kilogram Go ahead.
1 gram.
1 gram.
The protein is $80 million US. So this is based on the GFI.
80 million or 8 million?
80.
So what's costing $80 million for a gram? What component are you talking about?
So this is the TGF beta.
I'm sorry?
The TGF beta. So it's a transforming growth factor beta. And so this is a family of growth factors. And yeah, so they have TGF beta 1, TGF beta 2, TGF beta 3, and also bone morphogenetic protein within the superfamily. And they are quite difficult to make because all of the competing systems, they have been struggling with achieving high yield. And on top of the high yield, they also have a very complex biological structure. They have disulfide bonds within them. And then the process of making a functional protein is costly. So to keep it like simple. So this is the thing that we've managed. So in our case, our plants are producing large amounts of these proteins. So we're talking about 2 grams per kilogram of leaf material. And then we need to convert this amount that we are generating within the leaves into active proteins. And this is where we managed to significantly reduce the cost of manufacturing it.
So who currently manufactures the product that's $80 million for the gram?
So it's R&D systems.
R&D systems.
Yeah.
So does your product make their product obsolete or?
Yeah, well, obviously, if you look at the market of growth factors, they are usually sold in very, very small quantities because even for the cultivated meat sector, most of the companies now, they are at pilot scale. So they're only using micrograms amounts of these proteins. So you can go into the market and then you can see that the 10 microgram is sold between, 10 micrograms is sold between 200 pounds all the way up to 800 pounds, depending on the manufacturers, depending on the quality of the protein that you are producing. So and then if you want to order like larger quantities in milligram quantities, you need to ask for a quotation because it's not readily available. So this is the main problem. They're producing it at very low yields, and then they're not readily available. Now, according to the GFI report, if you want to produce 20 tons of cultivated meat, you need to have you need to use around 41 grams of growth factors, and then it's not easy to get those 41 grams.
So for you to create 41 grams of growth factor would cost how much?
So to make 41 grams, so it's 100,000 times, I'm very bad with numbers, 100,000 times 41. It might be very easy. It's an easy calculation. I just don't say a wrong number.
Okay. Don't worry about it. But basically, this is an unlock, certainly for cultivated meat. And it's potentially an unlock even more so, in addition, for regenerative medicine, right? Yes. This is as the ingredient is currently being used in both areas. If you guys can create it at significantly less, it's important. It's an unlock.
Yes. Yeah. So I think this is what we're offering. Like we want to produce these in large quantities so we can unlock and then allow this industry to scale up. This is the USP that
So it sounds like there's a regulatory component that you guys are going to need to go through. Obviously, there's the scale up of production. What do you think are the most challenging aspects of what you've got to accomplish in the next year or two?
Okay, so the first one is regulation. So and this applies to all of the manufacturers of growth factors to the cultivated meat sector. So it's not something that only Bright Biotech has to take care of. So we need to answer the questions whether it is okay to use growth factors from a genetically modified source. Now, for example, we've spoken with Food Innov in France. And then one of the things that we've discussed is like having a consortium of different stakeholders in the cultivated meat sectors that involve people producing the growth factors, people manufacturing the cultivated meat, and then answering the questions and in order to raise it to the EU Commission so they can look at it. But yeah, so at the end of the day, it's a genetically modified, or it's a growth factor from a genetically modified source, and it is not considered a novel food. So it might take a few years in order to get to this stage. So yeah, but then someone needs to take the initiative. I know, for example, Gourmet, they already included in their team a member who's been who's aware of the regulation and has been a regulatory advisor. So yeah, so it's these people that will allow us to clear the usage of growth factors from a GM source in the cultivated meat sector. So this is in terms of regulation when it comes to using the growth factors. Another thing, so in terms of plants, for example, in our case, since we are based in the UK, we're using tobacco plants. And then we're also using a chloroplast system. Our work is considered containment level 1. So it is the lowest risk. So there isn't anything stopping us from growing the plants as long as we abide by all of the rules and follow the health and safety regulations. So that's not an issue. So this is one of the things. Also, a third thing that we've already talked about is the nicotine content. So this is also very important. Now, there are other things that, for example, we don't worry about, for example, contamination from animal pathogens or bacterial endotoxins, and these are inherent disadvantages of the mammalian systems or microbial systems, which is now commonly known as precision fermentation. So in our case, we don't worry about these things because plants are generally recognized as safe, and then they don't carry over these contaminants. So this is when it comes to the when it comes to regulation. But it's something, as I told you, it has to be addressed by all of the different players in the cultivated, and we are one of those players. Now, other challenges that are related to our technology. It's not necessarily a challenge, but it's something that we'd want to address in order to expand the horizon of the proteins that we can produce in our system. Chloroplasts. They don't carry out all of the modifications that eukaryotic cells or animal cells can carry out. So because the chloroplasts, they, according to evolution, and it's a prokaryotic organelle that invaded the plant cell and it thrived inside. So basically, it has features of a prokaryotic system, which is the simple, like bacteria, is a prokaryotic, and then eukaryotic, which is like animal cells and plant cells. So it can carry out some modifications, and then there are certain modifications that it cannot carry out. So one of the things that it cannot do is something called glycosylation. And glycosylation is a post-translational modification that sometimes is essential for activity, and it's also essential for stability of the protein. So in our case, luckily, the growth factors are non-glycosylated, or at least the ones that we are producing and the ones that are of interest to the cultivated meat sector. And even if they are glycosylated, it's not needed for their activity or their bioactivity. But then there's always ways of optimizing the system and engineering a pathway of glycosylating or adding these sugar groups to the protein in order to make them active or stable. So this is a technical challenge that we can always think of to enhance or advance our technology.
In terms of sort of validating the effectiveness of your growth factor, do you need a certain quantity? I mean, are you guys looking to create a certain quantity that you can then give to companies, trying to work on cell lines? Or I'm wondering how you go from where you are to, and maybe you've already done this, go from where you are to producing a certain volume that you can then go test with other companies to see whether you're getting the efficacy you're looking for.
Well, at the scale the cell-based meat industry is at now, we are already producing a lot of the protein that we can start sending samples. So anyone who's hearing us and they are interested in growth factors, just reach out to us. And then we will happily send the growth factors that we have for them to test on their cell lines. And then if they are interested in any other growth factor that we haven't yet included in our production pipeline, please let us know about it. So we already have plenty of protein that we've manufactured, and they are stored at -80. We've already spoken to a number of cell-based meat company, and that was the significance of Big Idea Ventures, that they've connected us to them, and then they've expressed interest. We've agreed to send the samples. The thing is, they are at different stages. Some of them, they will start the testing later in the year. Others, we wanted to send samples to Super Meats earlier, like in December, but then because of logistical issues of sending samples on dry ice, it has been delayed. But then the samples are ready, and then it's just like having now the time and just addressing all of the obstacles for moving biological material between countries.
Got you. Are there different kinds of cells that you believe your growth factor will have either more or less impact on? So, for example, bovine versus marine versus, whatever.
Yeah, well, no, we don't have the answer yet. Actually, this is one of the things that that we will address very soon. So just recently, we added a new co-founder to our team. Rania Dirani, so she's been in the tissue engineering industry for so many years. She was trained in the US, and she joined us in November. And then one of the responsibilities that she will, one of her responsibilities is to set up a system for validating our growth factors on species-specific cell lines. And that way we will be offering our customers with protocols so basically, our growth factor will be tested on the cell line that they are interested in. Because most of the time, all of the growth factors that are currently available on the market, they are usually tested on mouse cell lines. So, and then they need to do a lot of optimization once they acquire those growth factors. So in our case, we want them to receive protocols that are specific to the animal cell lines that they are working on. So this is something that we will be developing in-house. So answering the question whether we know whether the one that we have now works better on a beef or a porcine, it hasn't been tested yet. This needs to be yet tested. But we know, for example, the protein that we have, it works so well on adipocytes, and these are fat cells. Because the cultivated meat is just not muscle cells, it's muscle cells, fat cells, and a number of other cells. So we know that it enhances proliferation of fat cells. So this could be very interesting to people that are looking at the other cells in a cultivated meat.
Interesting. Okay, fantastic. Given that you're based in the UK at this point, are you anticipating at some point taking your team or your technology to other parts of the world, i.e. US and etc.?
So we haven't put a block or we haven't ruled out moving out of the UK. Actually, just yesterday we were speaking with Genopole in France. And so everything is open. So ideally, we want a facility where we are in proximity to a glass house where we grow our plants and then we process the material. Also, if we're going to go with a partner in order to help us with the downstream processing. So we need to look at all of these things. So it could happen.
I'm not suggesting you move from the United Kingdom. I'm just suggesting that you have other places in other markets where you do what you do as well.
Yeah, well, remember we use plants, and then plants, they need a lot of light. And then if we're talking about growing them in the field, I don't know how favorable the climate in the UK is to grow them in the fields. But at this point, so I think the regulatory framework and also everything we need, so the open access laboratory and also Manchester, so we have excellent labs and excellent entrepreneurial ecosystem surrounding us. So we're happy where we are based right now, but then we're exploring other places where we can have this facility that includes all of the things that we need to have in place.
Perfect. So Mohammad, I've taken up more time than you probably promised me. So quick question. If someone's listening and wants to help you in some way, what kind of help are you looking for right now? And do you anticipate needing over the next 6 months or so?
So in our case, as I've mentioned, if there are any cell-based meat companies listening to us now, if you're interested in TGF-beta, which is the protein that we've got now, or any other proteins, including the FGFs and the IGFs, please reach out to us. We are more than, we will happily send you samples. So this is one of the things. Any other, for example, so one of the things that we are trying to do now is acquire cell lines in order to, as I told you, validate the growth factors in-house. So if anyone can help us acquire those cell lines in order to do the testing in-house, that would be great. And then the third one is we want to establish this infrastructure for global delivery. So also if anyone can support us with this, that would be excellent.
Perfect. Are you also looking for new team members, new funding, new funders, new investors, etc.?
Investors at this point, so we are very close to closing a round now. So it's just doing the legal work now and hopefully, announcing it or making it public. So, so if anyone is interested to listen to our story at this point and then start talking about Series A, so we're more than happy to. We're actually speaking with a number of VCs for Series A, involving them very early on with our technology and our company. So so this is in terms of investment. What was the other thing? Sorry.
Employees.
So in terms of employees, so now most of us in the company, we are of a technical background. So we want to expand the team to include people in communication. We want to include people also in finance. So it will, so in a few months' time, we will be advertising jobs that will involve like a finance position and also like a communication position.
So where do people find out about Bright Biotech? If people are interested in just finding out more about you, about the company, about your mission?
Yeah, so they can reach out to us on our website, www.brightbiotech.co.uk. So this is our website. So they can, so there's a contact form if they want to send us an email. Also, they can find us on LinkedIn. So it's Bright Biotech. And so they can also reach out to me on LinkedIn, which is Mohammad Khalil Hajj, Mohammad space KHALIL space EL space HAJJ.
Are you also on LinkedIn?
Yeah, so this is LinkedIn. So yeah.
Okay, so if I go to LinkedIn and type in Bright Biotech, you guys are going to pop up?
Yes, we should. We should.
I also just Googled it. Brightbiotech.com is currently for sale for $50,000. So I'm guessing somebody's sitting on that, or is that you guys?
No, I wish it was us. So from the day one that we wanted to purchase the .com and it was taken, so we went with the co.uk.
Damn, man. Maybe with the next fundraising, you go buy that puppy.
Exactly. Yes. And then they would increase the price knowing that we've raised money.
Yeah, well, keep that a secret until after you do the deal. Mohammad, I really appreciate your time today. We've had a number of conversations with cell-based companies, some companies sort of tackling the growth factor side of the equation, but this was probably the most knowledgeable interview we've had. Made me look ridiculous, but I don't care. That's what I'm here for. I really appreciate you taking us through it. I'm sure there are people here who will just kind of go up that learning curve just from listening to your conversation today. And they can always go do some more work after hearing you talk, or maybe they can reach out to you and ask you questions themselves. I don't know.
More than happy to speak to them if they're interested. Yeah, and thank you very much, Andrew, for having me on the podcast. I was actually looking forward to it, and I'm very glad that it happened, and I really enjoyed it.
I've seen you on stage. I can't believe you're an introvert. I've we've done the whole demo day in Paris. You standing up there and rocking the whole room of people. Yeah, you as an introvert. I never really put those two words together. Those two things together.
Yeah, no, definitely I am. And then this is what I've enjoyed about this learning process. And yeah, so I used to get exhausted from engaging with so many conversations, but now I look forward to meeting people and discussing what I'm doing and then hearing from them their feedback. So it's yeah. So I think I am still an introvert, but it doesn't exhaust me anymore engaging like and talking to people.
We're going to send you a T-shirt with like "I am an extrovert now" that you can wear around the lab. Okay, so Bright BRIGHT biotech.co.uk. Mohammad is the CEO and founder or co-founder?
Co-founder.
Co-founder. So he's offered to answer your questions, everyone who's listening, which is, I don't know, 5 people, my aunt, my uncle, my grandmother. I don't know who's listening. Thank you so much, Mohammad. I appreciate it. I'm going to press pause in a moment. And again, thank you for coming to the Big Idea podcast where we focus on food today.
Thank you very much, Andrew.
Okay, stop. And thanks for coming along to the podcast today. I hope you enjoyed the conversation with Mohammad from Bright Biotech. If you have any questions, please do reach out to us. Come along to bigideaventures.com or reach out to us via LinkedIn, YouTube, or one of the many places we're available. You can also reach out to Mohammad at Bright Biotech if you have any questions for him. Okay, hope you enjoyed the podcast. Love to get any feedback, questions, or comments you might have. And I'm Andrew Ive, the founder of Big Idea Ventures, here to kind of talk to you about some great companies on every podcast. So that's it. Look forward to hearing from you. Have a good day. Bye.